in starting today, the center of what we do, whether it's industry or the regulatory function or the innovative academic function, ultimately the patients are our goal, and innovation for them to address something really potentially terrible and multi-systemic like this. And so the voice of those people are something that really has to be central to what we do. And with that being said, I'd like to start off turning this over to Isabelle Lousada, CEO of ARC, to give us a patient perspective. And Isabelle, I'd like to thank you, A, for being here, and B, willing to talk about this, because I know you make it a habit not to do this.
So thank you so much, Preston. And while I rarely talk about myself, I'm very glad to be representing the patient community. So since the formation of ARC in 2015, and with the regulatory review of a number of therapies, one of the questions that's repeatedly surfaced around the choice of endpoints is, what does this mean for patients? And what value or benefit does this represent for patients? These vital questions have been raised by regulatory representatives, by industry, and by payers. And as Matt so eloquently put, patients want to know, how am I doing? So we felt it was very important as part of this discussion about the role, value, and potential of imaging to include patient perspectives. Last month, between October 20th and November 1st this month, we polled the patient community, and I'd like to thank the Amyloidosis Support Group, Muriel in particular, for sharing this with her patients as well as ARC patients. We had 653 patients who participated and took this poll. 37% with AL amyloidosis, 36 with wild type, and 20 with hereditary. Most, 64% of patients, were diagnosed 2 or more years ago. Changing my speaker. Of all patients, around 80% reported use of imaging for either their diagnosis and or monitoring of their disease. 90% of patients with ATTR wild type, imaging was used in their diagnostic workup, whereas for AL patients, it was about a third. AL patients, compared to those with ATTR, have more experience with imaging for monitoring their disease, and about a third of AL patients had no imaging. 19% of all patients reported having either a heart or kidney biopsy. Of those with imaging, about 75% reported having an echo for either diagnosis or monitoring of their amyloidosis. Experience with imaging as part of diagnosis is more frequent for those diagnosed within the last 2 years, 74%, versus 51% of patients who were diagnosed over 2 years ago. This is similar to what we've been seeing in the ARC community survey done earlier this year. Use of PYP appears to be increasing in patients diagnosed more recently, 58% versus 42%. Use of other imaging types has remained relatively the same. So it might be hard to differentiate on the slide, but the slightly brighter green seen on the right side of each line represents very important. Patients place a high importance on imaging, more so with the use of imaging to monitor versus diagnosis. When looking at the mean score of 1 to 5, with not important being 1 and very important being 5, the highest importance was placed on being able to see the impact of treatment with a mean score of 4.46, and importance of imaging and understanding disease status with a mean score of 4.43. All factors had a mean score of greater than 4. Patients also shared their personal thoughts, and some of the common themes are represented by the comments included here. It's less invasive than a biopsy. In some cases, biopsies have also produced false negatives. Some patients report insurance that will not cover the cost of imaging. And I think to Matt's point, this also relies on physicians sharing the data in a way that's accessible to patients. So my last thought is that coming to terms with a complex rare disease diagnosis and gaining vital information about the disease process and impact is challenging. Imaging can provide important information for a patient, a comprehensive view, if you will, of their condition and the impact of treatment. And I will hand back to Preston. Thank you.
Isabelle, thank you for this. And I'd like to proceed on from here with a case study from Dr. Masri, who's going to talk to us about a recent example that he has experienced using echocardiography as part of a development program. Dr. Masri is with us here from the Ohio Health Science Center. Dr. Masri.
Thank you. Thanks for the invitation. I do both HCM and amyloid, so that's most of my time spent both clinical and research. And we have had a lot of experience with Mavacamten as well as more recently Aficamten, cardiac myosin inhibitors in hypertrophic cardiomyopathy. So the point about obstructive hypertrophic cardiomyopathy is that it is considered a rare disease. It's actually common, but a lot of the times it's underdiagnosed or misdiagnosed or just ignored for a long period of time. However, even though we see a lot of patients with obstructive HCM, it is relatively difficult to enroll them in clinical trials, and there are accepted definitive treatments that transform these patients from obstructive to non-obstructive disease. And we have low event rate, which would require very large clinical studies with long-term follow-up. They are really impossible to conduct. And this is a classic figure used from this paper by Dr. Marin showing how the event rate is about 0.5% per year. There's a lot of controversy if this event rate is actually representative of the community event rate, but at least what we know is that it is low. And when we think about other surrogates, one of them that's been used lately is peak VO2. This is one of the papers that I worked on a while ago, looking at how when peak VO2 decreases by 1 point, it's associated with a relative risk reduction or relative risk increase by 9%. But prior to 2022, all the treatments in this space were based on observational data and expert opinion. There have been no medications which directly address underlying pathophysiology, and all of our treatments converge on essentially imaging evidence of improvement typically, because all of the data showing quality of life or difference in symptoms are observational. So this is the pivotal trial of Mavacamten. Mavacamten is a cardiac myosin inhibitor. What it does essentially is in HCM you have hypercontractility. Actin and myosin are interacting too much, and we want them to interact less, but not to a point where we can cause problems. And so this was a 30-week trial on treatment trial, and there was a washout period, a range of dose for Mavacamten, and what was looked at is this was placebo-controlled, and this was the primary outcome. It's not imaging, as you can see here. So the primary outcome was you either increase your peak VO2 by 1.5 and have an improvement in NYHA class, or you increase peak VO2 by 3 and have no worsening of NYHA class. This was the primary endpoint. And Mavacamten now is FDA approved for symptomatic obstructive HCM. When you look at the Mavacamten arm, 37% achieved this endpoint versus placebo 17%. This is important. Why? We're taking these patients who are obstructive, who have evidence of obstructive disease, and we're looking at a placebo effect of 17% using these surrogate endpoints. That, affects your, of course, effect size, sample size, all these things that you do. But look at the NYHA class improvement. A third of the patients said they felt better on placebo. And so this is the challenge of using some of the surrogates where we are looking at symptom only. assessing these patients. Now, compare that to what everybody thinks is the primary driver of symptoms in obstructive hypertrophic cardiomyopathy, and all of our treatment have historically been based on this measure, which is LVOT gradient. You can see the clear separation. Placebo is red, Mavacamten is blue, and you can see at rest, our typical threshold is below 30 versus Valsalva is below 50. And you can see how you can clearly take this imaging biomarker, which is the reason why we treat these patients in the community for the obstructive HCM if they have symptoms, of course, and we even do open heart surgery for them if they have symptoms for this reason. And you can see how the placebo group is consistent. There's some reduction over time, but it's not that impressive. And so when we talk about imaging as an endpoint in HCM, LVOT gradient is used as therapeutic target since the 1960s. Mavacamten has a REMS program. The REMS program is mainly imaging-based, which incorporates both LVOT gradient and LVEF. And all the safety analysis that's been reported so far is based on imaging, finding that the LVEF goes below 50%. It is not based on clinical heart failure. So the events that happened in the trial of interest were EF below 50%, although the majority of these events were asymptomatic. So that's another imaging kind of a marker there that we have been using. Now, LVOT gradient is not so far an acceptable target on its own for clinical trials of obstructive HCM. And there's a good reason for that, because if you reduce LVEF in a pathologic manner, you actually get rid of the gradient. So you might be harming people while achieving this endpoint. However, if you combine LVEF with LVOT gradient, there is no plausible mechanism known that you can actually cause harm or that you're not truly reducing LVOT gradients when your EF remains within the range that is acceptable as normal range. And feel and function can be enriched by these imaging. It's not a competition, it's not replacement, it's just essentially enrichment there. They're less prone to placebo effect. I'll show you the next slide. And then when we talk about the complex aging population, we need more tools that can separate why someone doesn't feel better. There are some, there are people we do surgery on, open heart surgery on. They come back in a couple of months and they say, I just feel the same, nothing changed. And so when we talk about also extending cardiac myosin inhibitors for preventing or improving cardiac structure in minimally symptomatic or asymptomatic patients, we can't rely on surrogates of peak VO2, of symptoms, or of hard outcomes. They don't have much symptoms to begin with. And so to move the needle, we need these imaging endpoints to help us in working these patients. And this applies to amyloid also, as the disease is changing in its natural history and what we're trying to do with early identification. This is just a summary of the placebo effect in Explorer. You can see how NYHA class, as I mentioned, a third. KCCQ is approaching the clinically acceptable threshold of improvement on placebo. So just give them placebo, they improve by 5 points. And so Mavacamten had 13.6 points there. But still, that's significant. That affects how we conduct these studies. Look at exercise LVOT below 37% only achieved that on placebo, probably from training effects over time within participation of the trial. Look at left atrial volume index, the last one, LAVI. This is associated with sudden death, associated with progression to heart failure, AFib. Placebo had no effect whatsoever on it, for example. And that's important. And finally, peak VO2, if you have a population that can exercise in a reproducible manner and don't have a lot of other comorbidities, which is not necessarily the case in amyloid, also the placebo effect is very minimal in this population. Now, what do we have on hand? We have VALOR HCM. This is the second phase 3 trial of Mavacamten, looked at patients referred for septal reduction therapy, which is either septal myectomy or alcohol septal ablation. The majority probably were for septal myectomy based on the centers involved. And the reason to say you need surgery is if the peak LVOT gradient is above 50, and which is an imaging endpoint, or if your NYHA class is 3 or 4. And so it showed that essentially you can prevent people from going to surgery or alcohol septal ablation if you give them Mavacamten. And this is now submitted to the FDA for review as to be added to the label of Mavacamten. But what I would say is that this is primarily driven by imaging. It doesn't matter what NYHA class is in the end of the day, because most people scale it up and down based on the day. But for peak LVOT gradient, Mavacamten will reduce it in a lot of patients below 50, and that would be a driver for a lack of SRT eligibility or becoming SRT ineligible. So that's another imaging endpoint there that could potentially become an indication in the future for patients with obstructive HCM. One other point to make is the primary endpoint was peak VO2, as I showed you. There were about a third of patients on Mavacamten who did not have enough improvement in their LVOT gradients. We actually, which we've termed non-responders, we actually don't know what happened to their peak VO2. So we want to see this analysis. We don't have it. I'm sure the FDA reviewed that analysis. But if patients had improvement in their peak VO2 without improvement in LVOT gradient, that's a problem, because we are all using LVOT gradient as our surrogate, but we don't know the results to that analysis yet. And other than a primary endpoint, imaging is used in every single step of the clinical trials of HCM. Disease definition is imaging-based, diagnosis is imaging-based, inclusion-exclusion is imaging-based, safety, efficacy, supportive measure of structural remodeling, and septal reduction therapy criteria. So every single step of the way we're using imaging in this disease. I'm not going to go over these two boxes, but this is almost copy-paste. There's a lot of shared and common features between HCM and amyloid. And I think HCM does present a very good case study to learn from and how we can apply things in amyloid. There's differences, of course, of the patient population, the age, the comorbidities, whatnot, but there are a lot of things to learn there, and imaging is one of them. Thank you.
Thank you for that. And one thing, we'll be addressing some of these things you just presented during the question session this afternoon. But one of the things that is very interesting here is that this concept that you don't have to jump straight to a biomarker that's a response marker, that you can get experience with something as a diagnostic marker, as a monitoring marker, as a predictive marker, not in the sense of predicting drug effect, but predicting how one population of people will behave relative to another, and whether the image is prognostic or not. All of that is important knowledge to gain before you take that last leap and say, okay, can we put this in an endpoint composite? And I think we're certainly getting there, but thank you for that. Okay, moving on then, we are going to go now to this part of the program today, where it's actually quite a treat. We have almost an embarrassment of riches in having regulators here from multiple FDA divisions, the MHRA, and EMA. And what I'm going to do as we start having this discussion this afternoon, understanding that what we want to do is hear from them and their thoughts about, this whole premise of developing imaging biomarkers, is how their particular division is looking at this, what they feel is important. Are there commonalities? Are there differences? And are the agencies looking at this in different ways around the globe? And so we've got, because of the number that we've got is really wonderful. What I'm going to do is start with FDA, and we're just going to go around the world. Okay. From what I understand, we've got the names of the participants up there. But what I'd like to do is, if we can, pull the virtual room up. Okay. Now, I see, just so I make sure I understand, I see the pictures of the people who are online. The people whose names are there, are they audio and not visual? They are. Okay, great. So you can see we've got quite a group here. And so what we're going to do is go around and give them both, time, say 3-ish minutes for to address these questions that we're bringing up today and see what their high-level thoughts are about that. Norman, I'm going to pick on you first. So if you get us started, that'd be great.
Well, this has been a very interesting meeting so far, and I appreciate you folks doing it here on campus with us. I think there's there are a lot of points that have come up through the day that I think are worth emphasizing. One is that when a biomarker is somewhere short of being ready to be a surrogate, there's still enormous value associated with using it for diagnostic purposes or using it to as enrollment criteria, using it to enrich a trial for events, or if you're very fortunate, enrich it for response to a particular intervention that you're trying to test. And because the bar for accepting something as a fully validated surrogate is a high one, I'm going to suggest to you that although I think it's extremely important to be doing the work that's going on here and trying to understand the measurement characteristics of the biomarkers and organizing a way to collect these data so they can be used for long-term purposes, it's probably not reasonable to lay out a plan that expects to get a particular biomarker validated for surrogacy. Because the surrogacy, we expect to see that regardless of the nature of the intervention, that the surrogate predicts the outcome that we're interested in. And you can't very well lay out a plan that's going to ensure that every biomarker goes through that sequence of interventions. So, there is a pathway. It does lead, maybe, to surrogacy, but the key things to do, or the things you're doing in terms of trying to find things that you expect to be disease relevant, and be very systematic about the protocols you use for collecting the data, the validation, which I think we need to have some further discussion about, and the basic infrastructure for keeping things together so that as experience accumulates, that you can build a case for a surrogate. I will also mention, and then I'll yield the floor, there is a step between uses for enriching trials and full acceptance as a surrogate. There is a potential pathway where things are, quote, reasonably likely in the United States. So that's the way the phraseology is, where something isn't fully validated, but there is a study that is predicated on demonstrating an effect on a reasonably likely surrogate, with the expectation that somebody will be able to do the trial that actually resolves the clinical benefit in the post-marketing setting. That works as long as you've got a pathway that somehow makes that post-marketing study viable. It's not a useful pathway if you won't be able to run the outcome study because event rates are too low or whatever. But that is a potential step somewhat short of getting something as a full surrogate. I'll stop there.
Okay. Norman, I just want to clarify for the group, when you're talking about this issue of surrogacy that may cause difficulty here with imaging, needing to basically qualify the image for multiple different drug combinations that would be the interventions of different clinical trials.
Yeah, presumably, the pathway to getting belief that this is a valid surrogate, it has to do with demonstrating that regardless of how you have altered the biomarker of interest, you get the same predicted outcome change. So you can't do that with one intervention. You need drugs of different classes, or drug and device that leverage different approaches. It's that kind of thing.
Right. And building off that, I would actually like to thank Dr. Gormley for joining us from oncology. Thank you so much for being here. Your timing was perfect because I suspected that's what we were going to hear from the cardiac side. Nicole, we'd love to hear your thoughts on this subject as well from an oncology point of view, understanding a couple of things. First of all, this group is very well grounded in both BEST and the PRINCE criteria. We've talked a lot about it. Everybody knows this is not a proprietary discussion today, and that you all are giving us your thoughts not on things you see today, but what we need to be doing in the next 5 to 10 years to bring what you'll need 5 to 10 years from now for this, because it's going to take us that long to do it. One thing in particular we're interested in is the subject of qualifying an image as a potential surrogate when the drugs are all different in the background. And would the meta-analysis approach somewhat address that as far as trial level predicate data, where, because, in cardiology, we've got this luxury of having huge trials, and we can say, okay, we're going to use that one as the predicate clinical data to compare and see what the biomarker did. Generally, your trials are not 6,000 people. And what I've seen, if some of them, they're 25, 50, that kind of range. And so to address this issue of would the image be suitable across drug classes, would a meta-analytic approach to the predicate database be something that we could do?
Thanks for the question. And hi, I'm Nicole Gormley. I'm a hematologist and the division director of the Division of Heme Malignancies 2. We oversee products for multiple myeloma, amyloid, and all the lymphomas, CLL as well. So, and I'm also the acting associate director for oncology endpoint development within the Oncology Center of Excellence, as I've had a significant interest in endpoint development and its use in drug development. So Thank you for the opportunity to be here today, and thank you for all the great discussions. I was glad that I was able to join from online and run over here in time for the in-person session, or my portion of the in-person session, but it's been a really good meeting. So I just want to, say that I underscore as well that I think it's really important, and I think it's really valuable to have everyone work together. These are very challenging. It's very challenging to develop biomarkers for potential endpoints, and it really requires a large group collaborative effort. So I applaud what you're doing. Just to start off, I will say that from an AL amyloid, perspective, we're coming from a slightly different place, right? In that, it's the underlying plasma cell dyscrasia that's sort of leading to all of this. And so I think we've had, some discussions in previous meetings as well. I think imaging has a potential to be really helpful and clinically meaningful in drug development. But I will say that I think as an endpoint, it probably needs to be evaluated in components or in conjunction with other assessments. Specifically looking at the, serum immunofixation and other, free light chain ratios, in addition to, other aspects. So in AL amyloidosis, I see it's pretty challenging to envision that it would be an endpoint in and of itself. Just, fundamentally. That being said, I think there are significant roles for it to help expedite and be useful just in drug development. We talk about biomarkers being useful for a variety of things, including patient selection, enrichment in trials, patient stratification, possibly being used to guide treatment decisions, escalation, de-escalation of care, and then as well as a potential endpoint. But those different uses that I just outlined have increasing risk, such that there's less risk of reliance on the biomarker from both the patient perspective and, from a trialist perspective. There's less risk with using it for patient stratification or selection than there is in using it for an endpoint. And the data required for each of those is different and increasing in scope as well. So, in oncology, we have, as Preston mentioned, several rare diseases, and then also, so some of our trials are kind of small, and then we also have on the other spectrum, and not necessarily other spectrum, but another issue that we run into is that we often have some diseases that are very indolent in nature, and so, reliance on some endpoints like overall survival in disease like CLL or follicular lymphoma or CML is very challenging. And so we have had a lot of history of development of earlier endpoints that can be used to make regulatory decisions. And in general, it is really important. One of the methods that's been used, it's generally thought, that the Prentice criteria are too stringent. And so there has been a reliance on other approaches, such as the meta-analytic approach, where the data are combined together and specifically using patient-level data to look at both patient-level surrogacy and then also trial-level surrogacy to see whether or not this can be potentially used as a potential endpoint. And, and that it's important when you're looking at a meta-analysis that ideally, that you would have, these should be derived from randomized trials with patient-level data to ensure that there's consistency of the endpoints and how they're defined within the various data sets. And then ideally there would be a wide breadth of trials, such as hopefully you would have some positive trials, some negative trials, such that you could have a greater definition of the specific relationship between the earlier endpoint and the ultimate long-term endpoint of interest. And so, it is feasible, and one other consideration that you have to keep in mind is as you mentioned, the therapeutic modality, such that that's used or included in those meta-analyses. And, that's something we engage in groups with multiple times when we're having these discussions, in that sometimes if a meta-analysis only consists, for example, of cytotoxic therapies, it might be difficult to extrapolate those results and use that endpoint when you're looking at immunotherapies, for example, or cellular therapies such as CAR T cells. So there really are a lot of considerations that go into looking at or considering use of a meta-analysis, and we encourage and interact and engage with those that are, conducting these sorts of analyses on a regular basis to really try and hone in on those specific details. But lastly, I would just sort of echo what Norman mentioned earlier. It's just that I think where, there's the most gain and one of the biggest problems that we sometimes run into when we're starting to look at the meta-analysis, the biggest gains that can be done right now is just making sure that the trials that are conducted in this space are looking at these biomarkers and imaging techniques in a standardized way across trials as much as possible, such that ultimately when you do go to do a meta-analysis or any other sort of evaluation, you actually have a robust data set. We found that sometimes, if they're done at different time points or they're done at different, assay types or, different thresholds, it can be really hard then later on to sort of collate that data and draw any meaningful conclusions. So, to the extent possible, making sure that these are incorporated in the ongoing trials currently such that you have the opportunity to learn from the data that you have now is one encouragement I would make. But thank you very much.
Just one quick follow-up question. In thinking about surrogacy of an image, is the counterpart to your last comments about drugs that the platform generating the image may produce variability if the platforms are different between your meta-analyzed data sets?
Yeah, I mean, generally, yes, it can. I think there's sometimes even, variability within, a particular platform just on, differences in how the information is collected, the sensitivities of each, type of, imaging, et cetera. So there are differences, and those sometimes you can overcome that, but sometimes you can't, So I think it does sort of require some thoughtfulness up front, to the extent possible.
Thank you. I'm going to just let everybody know in advance, because we've got this treat of having these people here today, we purposely put the 1 hour of discussion last so we could actually push into it if we need to, because we really want to hear what you have to say, so we won't be rushed. Okay, next we'll still at the FDA. I'd like to go to August Huffling. He's the Deputy Division Director of Imaging and Radiation Medicine here at Cedar. And so, August, we'll turn this over to you.
Hi, thank you. Are you able to hear me?
We can. Loud and clear.
Great. I just some brief background on my division. We regulate imaging agents such as contrast and diagnostic radiopharmaceuticals as drugs in the same manner as the therapeutic drugs that are regulated by cardiology, oncology, and other FDA drug review divisions. And I'm a radiologist. I'm not an expert in systemic amyloidosis or even its imaging, but I do appreciate the opportunity to contribute to these important discussions regarding relevant imaging endpoints. So first, I'd like to reemphasize the imaging standardization discussions from this morning's sessions and some of the related comments from my FDA colleagues. Standardization and related technical validation are very important for reducing variability and ensuring interpretability of imaging results. And optimizing imaging data quality, it's really as important as validating the clinical meaningfulness of an imaging use in therapeutic trials. And I think it's important to note that use of imaging in therapeutic trials will typically benefit from more stringent technical validation and standardization than use in clinical practice. And so I'd like to refer to the 2018 FDA final guidance for industry titled Clinical Trial Imaging Endpoint Process Standards. This guidance is very relevant. It highlights technical aspects of imaging endpoints that should be considered to optimize their use in trials. For example, in addition to modality, we need to consider things like image acquisition techniques, image quality control measures, and imaging interpretation methods. Of course, if we use imaging drugs, that adds additional technical considerations. And also, as discussed this morning, quantitative methods for imaging assessment of disease may be very useful and also bring additional considerations for technical validation and standardization. For example, the FDA held an entire workshop yesterday that was focused on just the technical issues related to quantitative PET for brain amyloid imaging assessment. So I'm not trying to add additional challenges, but just emphasizing that demonstrating the clinical utility of an imaging endpoint may be difficult if there are underlying problems related to its technical validation and standardization. I'd also like to make some brief general comments regarding use of imaging drugs for assessing systemic amyloidosis. As we know, imaging drugs have not been FDA approved specifically for this use. And an imaging drug may not be necessary for use in amyloid therapeutic trials, for example, the use of non-contrast echo. However, if imaging drugs are used in these therapeutic trials, there is, as you know, potential for repurposing of approved imaging drugs, such as GBCAs, bone scintigraphy agents, or brain amyloid imaging agents. And the established quality and safety of approved drugs are clear advantages that could benefit standardization and adoption of imaging drug use in therapeutic clinical trials. On a related note, it's possible that parallel development of imaging drugs and therapeutic drugs could be considered. We've seen a similar approach in the oncology space. Uses of imaging drugs as therapeutic drug development tools may also have general clinical imaging utility. For example, uses for patient selection, prognosis, prediction of treatment response, monitoring for disease progression or treatment response. These uses may all be useful not just for clinical trials, but also in clinical practice. So it's actually worth considering collaboration with imaging drug manufacturers, which could potentially catalyze imaging endpoint development. Those are my comments.
Okay, I just want to make sure we didn't cut you off there. One thing that's curious to me, August, if an imaging modality comes into FDA, who regulates that? Does it go to oncology if it's the drug being tested is an oncology drug? Does it go to cardiology if they're doing this as part of a cardiac trial?
No, so if it's,
who gets this?
If it's an imaging drug, so a contrast, like CT, MRI contrast, ultrasound contrast, or a diagnostic radiopharmaceutical for SPECT or PET, or even a fluorescent optical imaging drug that we're starting to see a lot of. So if there's an imaging drug involved, that is regulated by my Division of Imaging and Radiation Medicine, which is in the Office of New Drugs in Cedar. If it's a modality, like you say, if it's a non-contrast technique, a new scanner, or I suppose, potentially even a new
Hi, could you please
imaging
describe
device
your outdoor seating option? I'm looking to make a reservation for tomorrow around 5, and I see you only have
Sorry, hearing some
outdoor.
We've got
Is it
somebody who's not on mute. If you could mute yourself, that would be great.
If it's a new device, that would be regulated by CDRH.
Very good.
But
Thank you so much.
Just to clarify on your question, we may consult the therapeutic divisions when we have a new imaging drug, but the overall the regulation is within the Division of Imaging and Radiation Medicine.
Got it. Thank you. Okay, and then so now we'll go to Joseph Rajendran, who's a medical officer here also in Division of Imaging and Radiologic Medicine. Joseph, are you with us?
Yes, thank you. I am with you in full force. Thank you. And I fully agree with what Alex Hoffling has mentioned. It's important that as we develop these imaging methods as possible surrogates at some point, we need to have, first of all, have the imaging agent or the modality validated. So what we are expecting, whether it is doing the expected job or not. In other words, you're validating the diagnostic agent first, and then we will see how it will relate to the clinical situation and the progress of disease and whether it can work in that way. And it may or may not be possible to have a truth standard in all patients, as we have seen with the endomyocardial biopsy, which is a difficult situation. But there are always ways to go beyond that. It may not be possible in every case. But the other thing also is to see that whenever we talk about surrogate, we are expecting that it will be like the truth standard, but it may not always be the case, and we may have to take help from the other imaging modalities. And the other thing is what we see in one organ or one region may not be reflective of the whole process, as we have heard repeatedly this morning, is that what goes on in the heart may not be reflected or may not be reflected exactly in the same manner as in the kidneys. So wherever possible, amyloidosis being a systemic disease, it is important to look at the whole body, so that we will know what exactly goes on. And the other thing we talked about is about the role of POIP, the technetium bone imaging agent, and which is a non-specific agent, and we still do not know how it works. But the one thing that we will have to see here is, people have expressed that the changes may not reflect the changes that's going on in the body, which is true, because some of these bone agents go into the so-called third space, like pleural effusion, ascites, and things like that. So the changes may be, we have to look at all these things very carefully. I believe that's what I wanted to kind of convey. Thank you.
Well, Joseph, thank you so much. And if there are other comments, we can get to them in our discussion section later on this afternoon. Okay, next, I'd like to actually turn this over for comments to Dr. Rosalyn Adigun, who's to my left here, who is CDER's liaison to this public-private partnership.
Thank you, Preston. And I want to say a big thank you to everyone who's here today. So I work in the division of cardiology and nephrology products, so I second Dr. Stockbridge's comments, who is my division director. But I wanted to take a slightly different approach and just offer a few comments and segue to a slightly different area that I think will be helpful with this public-private partnership and in our understanding of the different regulatory gaps in amyloidosis as we go forward. So one of the unique features or one of the unique strengths of imaging is it interfaces at different time or different places in the medical product development sphere, in as a diagnostic biomarker, prognostic biomarker, monitoring biomarker. And I think as we learn from each other, one of the things that is important and will be emphasized by my colleague who is also here from the Office of Rare Disease is how we need to leverage all of the resources we have available in this room going forward to increase our understanding of these imaging biomarkers, leveraging their strengths and trying to resolve the uncertainties that we've identified so eloquently today. I think imaging is very important in protocol development from a regulatory standpoint as a regulator in study design, screening, inclusion and exclusion criteria. And as we review regulatory submissions in our understanding of imaging charters, I think we've emphasized the importance of standardization. And ultimately, as Dr. Masri emphasized, in treatment and follow-up, a lot of these imaging modalities are used to follow patients up after the drug has been approved. So I think part of what we would hope to accomplish after today's over to identify ways that we can all leverage what we have developed or understood in our different institutions to help advance what we know and how we can focus moving forward in amyloidosis. So what I would like to do is invite my colleague, Dr. Cynthia Welsh, who is with Office of Rare Diseases, because one of the things that we have been looking for or think would be helpful is trying to identify ways that we can leverage the data that is available in individual institutions to understand how best we can validate the imaging biomarkers that we've identified today as surrogate endpoints to advance the area of amyloidosis. So, Cynthia, are you there?
Hi, good afternoon, everyone, and thank you for including us. I wanted to point out some of the ways that the FDA has programs to facilitate endpoint advancement. And one of the new issues that we have is called RDEA, which is the Rare Disease Endpoint Advancement Pilot Program. And it's for developing novel endpoints in rare diseases, because we know it can be challenging. And we have this new pilot program through the PDUFA 7 commitment. And it provides a mechanism for sponsors to collaborate with the FDA throughout their endpoint development. To learn about it, we have a website that I can put in the chat where you can learn more about the RDEA program and how different sponsors could apply. Another program that we would like to discuss is some of the difficulty with data sharing, data use agreements, and a lot of these problems are the same across all the different development programs for rare diseases. And it's very important to know that splitting data is detrimental to drug development programs. And we encourage all stakeholders to share, and the pre-competitive space is the best opportunity to come together and share that data. There's the Rare Disease Cures Accelerator Data Analytics Program, and it is a program where they aim to facilitate cooperative approach and common standardized platforms to better characterize rare diseases and incorporate patients' perspectives in clinical outcome assessment measures and to build clinical trial readiness in the pre-competitive space. And I can give you links in the chat for a recent article that was written by the Critical Path on data sharing and a recent webinar from RDCA Depth. Another program that we have that's new is ARC, Advancing Rare Disease Cures. And the vision of CDER's ARC program is speeding and increasing the development of effective and safe treatment options addressing the unmet needs of patients with rare diseases. And the mission of CDER's ARC program is to drive scientific and regulatory innovation engagement to accelerate the availability of treatments for patients with rare diseases. With over 7,000 rare diseases and conditions that are very complex, and endpoint selection is particularly complex, particularly if there is a limited understanding of the natural history of the disease. CDER's ARC program is a CDER-wide effort that includes representations from the Office of the Center Director, Office of New Drugs, Office of Translational Sciences, and the other CDER offices, particularly in O&D, as needed for specific subject matter expertise. And there's a website that I can also put in the chat for you so you can learn more about the ARC program. And we really use that program to support sharing of learning about rare disease drug development and design. And there were 2 conferences recently that were held this year. One was a translational science in drug development, surrogate endpoints, biomarkers, and more. And CDER had an NIH, NCATS, FDA regulatory fitness workshop. And I can put both of those links in the website as well. And for the FDA NCATS regulatory fitness program workshop webpage, if you scroll all the way down to the bottom, there is a document entitled FDA Drug Development Resources for Rare Disease Community and More. That has a number of guidances and a variety of different resources that are useful for endpoint development and early drug development in rare diseases. Thank you.
And Dr. Walsh, thank you so much, and we really appreciate you being willing to share those documents with us in the SharePoint or in the chat, and we'll certainly make sure everybody has access to that. And thank you again for doing that and for joining us today. So next, what was to do is now go to the UK, and where our colleague Krishna Prasad, who's a physician at the MHRA, will share his comments next.
Well, thank you very much. Can you all hear me? I just wanted to confirm.
Loud and clear.
Yeah, thank you. Well, firstly, thank you for inviting me to participate in this. It's been a fascinating day, fascinating morning to hear various thoughts about what the markers should be and what the disease itself is and the multi-system nature of the disease. So I'm going to try and limit myself to two areas of what I can comment on. One is the various aspects of cardiological manifestations in the context of a multi-system disease, and where do we take the markers which address the question that we were asked earlier about the background fatality rate and whether we can identify an imaging marker. Having heard the various components of multiple markers and their functions that we have listened to and we've had experience of over the years, in my mind, I think to develop a single imaging or a surrogate marker for a multi-system disease is quite a complex experiment to conduct, and it requires a lot of data acquisition and efforts, including clinical trials. So then comes the question of whether we can use a single marker or a single modality for clinical trials only, but that is non-representative of clinical practice, and wherein multimodality imaging and multimodality images are used for decision-making process. In that context, I'm conscious of the number of echocardiographic and CMR parameters which are put forward, and which one would be the ideal version to develop. And all of them, as we have heard all morning, do require further efforts to defining the relation between the patient expectations of the disease and patient expectations of the biomarker and the regulatory expectations and the clinician expectations. In order to triangulate all of those, I think would be quite an arduous task. And as I agree with, as Norman has pointed out, that this is something which we have to identify, to clarify in our own mind as to what is the direction of travel that we wish to achieve at the end of a particular amount of time. Going to the multimodality disease, if we choose a particular imaging modality or if we're an imaging marker, is it sufficient to address the various components of the multi-system disease, and whether we can rely on just one component to say this is representative of the entire disease itself? Then comes, in my mind, comes the potential for various interventions having different effects, and can we find a marker, as Norman asked earlier, that will it be independent of the interventions that we plan to? Within the MHRA, I think when I got the invite, I did try and gather some of the information as to various colleagues from various expertises. And we did find it difficult to just come up with one solution or even a series of suggestions. The potential question that, or potential solution that I was offered was, can you use a multi-marker or composite multi-marker to take this forward? And I didn't hear much of the discussion earlier today, and I think maybe that's another component that we can explore. The validation of the markers is a difficult path, and I think we have all learned that many of these markers will get validated simply because they're used in clinical practice and there is no regulatory pathway for them. But it is good to identify whether that is an acceptable path that we want to go forward. It may take a long time in deciding whether that is an appropriate method, but I think that's something which we could consider using this forum such as this to agree quite rapidly across the various organizations and the stakeholders to come up with a process. Whether that process is, how long it will take and how successful it will be is a good question. Colleagues just now from FDA have talked about the rare diseases, and I think there is an element of learning from the smaller clinical trials in various other disease areas. And in cardiology, I think it's a victim of its own success of it being able to conduct large clinical trials, but even for defining surrogacy. So I'm not entirely sure I solved the problem, but I think those are my thoughts at the moment. I'll stop here and take any questions.
And if anybody has a burning question for Dr. Prashad now, we can ask him now, or we can, his time is late, and so he may not be able to stay with us for the whole discussion. So if we have questions for him, we can, now is fine. Okay, if somebody thinks of one, we're also going to stay on the continent here. So if you think of one, let me know. And so we'll go now to Clemens Mittman, who is with the Federal Institute of Drugs and Medical Devices in Germany. Dr. Mittman, can you hear us?
Yes, I can hear you well. Hope you can hear me also well.
You're loud and clear. And thank you for sticking with us at this late hour.
Okay. Thank you first for the invitation and for the great presentations, the high quality presentations and discussions here. I'm from the Federal Institute for Drugs and Medical Devices in Germany, from the Cardiology and Diabetes area, and I'm at the EMA and the Cardiovascular Working Party and the Scientific Device Working Party. There's a very formal question about the acceptability. We have a procedure at the EMA for qualification advices, where if a company, a consortium, wants information about whether a procedure, a method, or what else is ready to be used in a specific context, they can come for qualification advice, and then it will be discussed in depth. And we have sometimes we have procedures on markers that go for secondary endpoints or primary endpoints, and it is a dialogue procedure, and this procedure is often fruitful for both sides. And there are examples where a marker started at a secondary endpoint, at the end made it to a primary endpoint. So that's the formal the formal answer. The contemporary answer is that drug development, in my opinion, is not only about the phase 3 trial. It's not a one an all or nothing question whether a surrogate or a biomarker can be accepted as an endpoint, but it depends very much on the context of use. The hurdles, I agree with Norman here, for to be used as a single primary endpoint in the pivotal phase 3 trial are high, but there are different uses in the drug development. The hurdles are lower if imaging methods are used for proof of concept, if they're used for dose finding, if they are used for extrapolation of data from one population where we have data to another population with very few patients where we never will be able to get more robust data. For instance, patients with rare diseases with rare amyloidosis instances, hereditary disease. So if imaging data can be used for extrapolation issues. So there are different uses within the clinical program, and imaging data used for proof of concept can support other clinical data for 6-minute walk test or quality of life, and contribute to the overall package when assessing benefit risk of a drug based on the totality of data. Regarding use as a single primary endpoint, either one imaging information or in a more complex way with different imaging data going into an endpoint, the hurdles are high. We need to validate that for specific patient population. it has to be clear that if we have a correlation between the change in imaging for one drug for stabilizer, does that also hold true for a silencer or for a drug that improves that improves different agents. So the transferability of the results from one to another drug class has to be shown. If we have used cardiac biomarkers, is that how representative is that for the whole disease, for aortic stenosis, for thromboembolism, for conduction disorders, atrial fibrillation, or involvement of other diseases for the totality of the disease state of the patients, and then benefit risk is about quantifying the benefit for a patient in relation to quantifying the risk. What does it mean to a patient if extracellular fluid or extracellular space is improved by 20% in terms of patient benefit? That has to be defined, and therefore, as has been said, to be used for a phase 3, the hurdles are high. Last point, if it comes to accelerated procedure or conditional marketing authorization, that is the procedure we have in Europe, that depends in part on the drug. There are different requirements. At the time of the conditional marketing authorization, the benefit-risk balance also has to be positive, and that requires a sufficient validation of the endpoint. You need an unfulfilled medical need, and if you come with a drug where we also already have one that is licensed with the same mechanism of action, the medical need will be lower. And something that is also we always have to take into account is if we have a drug on the market license based on conditional marketing authorization, that automatically has an impact on whether we can generate further data later on. Patients may not be willing to be included or stay included in a controlled trial, so conditional marketing authorization may have a negative impact on the data that we get later on. All of these things have to be considered when using imaging data for conditional marketing authorization. It is evaluated by the EMA, so no general comment can be done provided on that. So thank you again for giving me the opportunity to share some thoughts.
Dr. Mittman, thank you so much for sharing that with us and for sticking with us at this late hour. I think that Jean-Michel Race from the French agency had to drop off. Am I correct about that? And so not hearing from him, I think that was the case. And so what I'm going to do is just thank every single one of you that came today for making time for this and for this group to hear what you had to say. We are very aware of the demands on your time and what you gave up doing today to be here with us, and really appreciate it. So with that, actually, I'd like to give our regulators a thank you. Okay, what we'll do here is proceed on then to the last session of this block, which will be a presentation of the NIH Heart Share Study and the Biodata Catalyst, presented to us by Dr. Vandana Shakdev.
Thank you very much. And I understand I'm sharing slides, right? So I'll be sharing my screen. First of all, I want to thank I want to thank you for the opportunity to be part of this discussion. It's been really engaging so far this morning, very informative. And what I'll be talking about is a heart failure program, so shifting gears a little bit. But I don't need to tell this audience that there's quite a bit of overlap and commonality between heart failure and the amyloid population. So So what I thought I would do is tell you a little bit about this heart failure program, the resources that went into it, and then we can talk about the overlap or the link between how this program might help with the discussions today. And I have two goals. One is that potentially describing this program to you might help you think about how it could serve as a model for amyloidosis research, particularly with imaging. And the second thing is it might serve as a resource for some of the work that has been talked about that needs to be done for imaging and amyloid. So I'll spend a few minutes quickly just going through exactly what this program is. Okay, let me know if you see this in presentation mode. But I'll talk about the Accelerating Medicines Partnership for Heart Failure called AMP-HF. And this is a public-private partnership that's managed by the Foundation for NIH, a separate entity that manages public-private partnerships. They've been creating these partnerships since 2014. There are several of them in existence on diabetes, rheumatoid arthritis, Parkinson's, schizophrenia, and even rare diseases with gene therapy. AMP Heart Failure is focused on heart failure with preserved ejection fraction, and it's the newest program. But really what it is this program started out as thinking about whether there is a biomarker ready for qualification. FNIH has a process where they create partnerships for diseases where a biomarker is ready for qualification. And for HFpEF, we realized after many discussions that we really don't have a biomarker ready for qualification or for immediate use. What we realized is we need to do deep phenotyping of what this disease is. As many of you know, we needed to deconstruct what the syndrome of HFpEF is. And so what we thought we needed was to do some deep phenotyping using all of the data here, clinical data, digital biomarkers, advanced imaging, and especially lab data and multi-omics. Tissue biopsy is critical in this. And we thought that using advanced analytics, we could potentially classify the different subtypes of disease. And then for those specific clusters or subtypes, we could identify new treatment targets based on the underlying biology of the different subtypes. Now, important in that, implicit in that is really that we need a biomarker or some sort of multimodal biomarker to identify each specific subtype of disease. There are two components to this program. It's a 5-year study with a retrospective component where we aggregate a lot of existing NIH and industry data, and we use multi-omics to define the different subtypes. And there's a prospective component where we create a new cohort of HFpEF patients. We develop ways to identify the different subtypes of disease and also look for targets in the prospective cohort. The goal at the end of this, of course, is to get to a precision medicine kind of approach like oncology, which was discussed earlier, to be able to direct treatments for specific subtypes of the disease instead of the one-size-fits-all approach. And so what resources went into this type of a partnership? As I mentioned, it's a pre-competitive partnership. It's a one-to-one between government and industry, and everyone gets access to all of the data. So coincidentally, the public part or the government part started about a year before the private part, just because of how funding decisions are made. And so NHLBI's program is called the HeartShare program. And we put out 2 RFAs for a data translation center, which was awarded to Sanjeev Shah at Northwestern, and 6 clinical centers will put together that new cohort of HFpEF patients. And we do have 2 HeartShare co-chairs as part of this program. The NHLBI has put in a lot of additional resources besides the 2 RFAs that I just mentioned. I'll talk about Biodata Catalyst in a minute. That's the most important for this discussion today, but we also have a top med program that provides omics for clinical research studies. And we have a lot of data in existence in our longitudinal cohort studies. So many heart failure patients within those cohorts, many HFpEF patients within those cohorts. So for this type of deconstruction of HFpEF, we needed a knowledge portal or a data platform. All of these large partnerships have in the past created their own knowledge portal, but because NHLBI several years ago had started to have its own cloud-based ecosystem called Biodata Catalyst, we decided that it made sense to use this for the AMP heart failure program. And basically what this is, it's a data platform that is cloud-based. It's used for data storage, data cleaning, data harmonization, and especially for data analysis. So it's not a download your own data kind of system. It's a place where tools are provided for analysis and tools can be brought in for investigators to analyze the data that's there. If you're familiar with the NIH other resources, BioLink and dbGaP, those were not cloud-based and they did not include imaging. The goal for Biodata Catalyst is to be able to create imaging repositories where we can actually do the machine learning, the deep learning, the AI with the images themselves, with the raw data, as opposed to just the measurements that were available in BioLink and dbGaP. So that's an important aspect of this program, as we know that imaging, it was discussed this morning, that AI with the echo images is going to be very important in the next few years, and especially for HFpEF, that's an important aspect of the diagnosis, and potentially it could be for monitoring and treatment response as well. And so the other part of this program is the private component, and these are our private partners. We launched the full program, the public-private partnership, just a couple of months ago on World Heart Day. And FDA has been an important part of this, with Dr. Stockbridge providing guidance and advice for several years now. And my colleague at FNIH, Tanya Camphouse, has been sort of the lead to put this program together. So if you have questions, her email is here. Just to summarize quickly before I get to the links now between amyloidosis research and this program, the NHLBI HeartShare program was launched initially about a year ago. We're putting together data from NIH cohorts and trials for the first component of this program, and we've set up 6 clinical sites to create a new cohort. And all of the data, as I mentioned, will be harmonized and cleaned and analyzed within Biodata Catalyst, and all of our partners within this partnership will have access to all of the data, and they'll have access to all of the images so that they'll be able to direct the research questions and do sort of combined analyses. The private partnership also now has been launched. And so I noticed Michaela from Ionis is here today. Michaela is one of our partners, and potentially they will also, the private partners will also be bringing in trial data, and we may end up using industry funds to either set up more sites or add to the deep phenotyping as the committee decides. So what are the links here, and how could we use this to either serve as a model for imaging research in amyloid, or to actually set up ancillary studies where we could use this huge data repository to compare and analyze with amyloidosis research. So to give you a little bit more detail on the retrospective portion, first of all, as I said, we're bringing together a lot of NIH and industry cohort and trial data and doing multi-omics to try to find the different subtypes of disease. We have actually talked about amyloidosis trial data, and could we use those data sets through some mechanism to compare with the existing data sets that we'll have on our existing cohorts and trials, a lot of heart failure data that NIH has. In the prospective portion of the study, now we're trying to deep phenotype this new cohort, but before we get to the bottom of this pyramid, which is the 1,000 or so patients that will undergo deep phenotyping, I'll tell you a little bit about the way that we're starting the more superficial phenotyping. What we're doing is at the 6 clinical sites, we're collecting all of the electronic health record data, which will probably be over 100,000 patients. And that data will be collected longitudinally to give us an idea of how much heart failure there is at each of the sites, what are the characteristics of those patients, what are the potential subtypes using just EHR data, and what are the clinical outcomes for those patients. Now, with all of that, that's all heart failure. And as I said, this program is mainly focused on HFpEF. So from that top level of the pyramid, we'll be reaching out to patients through a Eureka app, which is a platform some of you may be familiar with through UCSF. Jeff Olgin and his team have used this app for many clinical studies where they can reach patients for online consent. This is all remote. They can send them surveys. They can upload wearable data. And so what we have planned is to have a low-touch registry formed through this app by reaching out to HFpEF patients that are identified through the EHR data and to ask them to participate in this registry, where we will then, once they consent, send them surveys, collect information remotely just on HFpEF patients. Now, the reason for this registry is, again, to collect a little more information remotely, to collect information on outcomes, but and more importantly, to have a collection of patients that are interested in clinical trials and ready to volunteer for any studies that may come up. From that registry, we'll be looking for patients that are interested in undergoing deep phenotyping. Now, realistically, a lot of our new cohort will be selected and enrolled from patients coming to clinic, patients referred to our investigators at the 6 sites. But this low-touch registry will be one mechanism to find patients for this group, that about 1,000 or 1,500 patients that will undergo actual deep phenotyping at our 6 sites. It'll be 2 days of testing. It'll be cardiac testing. It'll be extracardiac testing, potentially, skeletal muscle fat biopsies, myocardial biopsies, right heart catheterizations on subsets of patients. And with the goal being that we're looking to identify what are the different subtypes of disease, and potentially after the 5 years of this program, can we do some sort of a master protocol where we can screen patients that have symptoms and signs suggestive of HFpEF, but then direct them into the precise therapy for each specific subtype of disease. Now, if we think backwards a little bit, someone talked this morning about, are we being too narrow in identifying amyloid patients with the imaging that we use? So ideally, if we were screening patients, we could identify amyloid and identify different HFpEF subtypes and direct them to the right treatment based on what was selected. Finally, before I finish, one thing I should mention is that with this HeartShare program, we are asking people that are interested to get in touch with us for ancillary studies, because the decisions for this program are made by the public and private partners together, but there may be amyloidosis is a good example. There may be situations where a lot of the infrastructure that we have could be used for different types of research. So, for example, if someone wanted to use the low-touch registry to look for amyloid or HFrEF patients or some other cardiomyopathy, that's one possibility. At the bottom of this pyramid, as we screen through patients that are eligible for the deep phenotyping, people that are diagnosed with amyloid will not be part of these 1,000 patients, but I'm sure that there will be patients identified with amyloid that could be sort of funneled into a different study, so to speak, if there was an ancillary study to do that type of thing. So I'll stop there. That was just a quick overview and some ideas for how this might relate to the discussions from earlier today, but I'm happy to take any questions.
Does anybody have a question for Dr. Stockbridge you'd like to put forth right now?
I have one comment and question. I guess maybe comments. Rick and I have been approached by Sanjeev, and we're happy to utilize the cohort that we're creating for SCAN-NP to include in this, which would be a cohort of patients, at least we hope 10%, who have amyloid and might be useful. I think the one thing that's also unique about amyloid, I would say, is that we're a little bit further ahead, I think, than HFpEF in the drug development arena. And what I fear as a clinician is that we're going to be faced with this difficult decision of how to pick which drug people get and what sequence, and we have absolutely no idea. And at the end of the day, I think because the government pays for most of these drugs that are pretty expensive, some ancillary study that enrolled amyloid patients may be very useful to clinicians, because if we really knew what was happening to them over time, we'd be better data-driven rather than, some other way about deciding what to pick.
Interesting comment. Matt, do we know what to do with patients who have HFpEF and an amyloid? Say the amyloid's diagnosed later, or do they respond well to SGLT2 inhibitors?
We don't know yet. My sense is they do, but what we need to know.
Any other comments?
Yeah, just one comment. So really excellent and informative presentation, Dr. Sachdev. The question I had was, there's been a lot of discussion on how patients who have HFpEF from amyloid may or may not respond to conventional HFpEF therapy. And in a registry, in a study such as this, the heart share with deep phenotyping, would you think it's important to exclude amyloid using PYP or something in these patients? Like, how do we know that the patients who funnel down have or don't have amyloid? Should we be proactively doing that? I'm sure you've had discussions. I just wanted to hear about your opinion.
Yeah, that's a very important question. I will tell you that one of the benefits of having the government part of this, the Heart Share study, funded a year before the program launched, is that it gave our investigators many, many months to talk about what that deep phenotyping protocol should look like and how do we find patients to get to that protocol. And that was an important part of the discussion is, should we be doing cardiac MRI on everyone? Should we be doing PYP on everyone? And as you know, funding is always a limitation, and as well as patient burden, that's an important limitation. So to make a long story short, it was decided not to work up everyone for amyloid because of all of the reasons I just mentioned, but it's an important point. And, you know, for using the clinical algorithms for which patients are high risk, I think that's pretty much what all of our investigators have been doing clinically for their other studies on HFpEF. And so that's the plan as of this stage is people that are high risk for amyloid that have suggestive features, they will get worked up and others won't. So it's not 100%, but it's the best that our group decided we could do.
Matt, kind of extending from that, in your institution, people who come in for an aortic valve procedure and have LVH, that everybody assumes it's because of their valve, do those people get screened with imaging for amyloid?
Yeah, Becky Hahn at our institution, when we did the Unveil trial, showed us all that if your S wave on your mitral inflow is on tissue Doppler is less than 6, that's 100% sensitive, so identifies low core, and then the group will usually not stand in the way of the TAVR, so the amyloid assessment occurs after the TAVR is done, but they will get an assessment subsequently.
I should add that there is a lot of interest in our group in using ECG and Echo AI to do that type of thing. And, you know, if there are algorithms that people are interested in testing, or if there are things in development that we could use to do that type of thing for HFpEF, we definitely would be interested. Matt, I'm glad you're already in touch with Sanjeev, but if others are interested, I'll put my email in the chat. Please feel free to reach out to me.
Oh, please do, and really appreciate you sharing that. Okay, well, thank you all for this last session. Really enjoyed this. We are going to end on time, so we will abbreviate the last section a bit to make sure we're finished on time. We are already getting some interesting questions coming in, and so look forward to it. Why don't we take a 10-minute break, and then we'll meet back and finish on time. All right, thank you all for sticking with us till the very end here. We've had some really interesting questions. One I'd like to address right off the bat. It came in from an online participant asking if you get 2 readings of a prognostic biomarker, one that you know is prognostic, and there's a drug-induced change, does that mean it's a response marker by definition? And I'd like to say absolutely not. That won't work. And let me give you a good example. In the heart failure literature, there's a drug that FDA pulled off the market long ago called flusaquinone, which was, I believe, a PDE3 inhibitor. And the outcomes all look good. People's exercise time went up. Their wedge pressures went down. Their cardiac output went up. They felt better. And they got full approval, but with a post-market requirement to do an outcome study. And as I recall from the details of that outcome study was terminated at the recommendation of the DSMB after 10 months because of increased mortality. And so correlations do not causation make. And when the BNP assessments from that trial were looked at, baseline to when it got, when it got stopped in follow-up, the patients' NT-proBNPs were actually going down while mortality was going up. Well, that would be counterintuitive in a heart failure trial with other agents. That you have this tendency to think if it increased your, lowered your your systemic vascular resistance and raised your output and all that good stuff it did, that, well, surely this biomarker would go in the same direction. And it didn't. It went in a way that was that would have suggested benefit. So that's as far as being prognostic at baseline, trying to impute from that or even a repeat of it at some point that you've satisfied all of Prentice's criteria, that won't work. So that was item number one. The other thing I wanted to touch base on in that regard, I've seen a lot of attempts coming in to use real-world evidence to supplement RCT data, either for an original approval using a hybrid approach in small populations or as a label extension exercise. When you're measuring a biomarker in the blood, what you quickly find when people go to registries to get that data, and they come up with a great result, but then you go back and point out that all those NT-proBNPs, for instance, in that registry were not only drawn not systematically, but they were all drawn from different labs that had different normal ranges, different performance characteristics, and none of them are relatable to the others as far as the performance characteristics of the test. And so my question to Dr. Dorbala that I wanted to get some input on is the same concern there for platforms in imaging real-world evidence.
I think that's an absolutely great question, and it really comes down to, within patient change. So if you're talking about imaging and you want to detect change, I think Dr. Rodney Falk and Dr. Sarah Cuddy also brought this up. Even if you use GLS as a marker, GLS absolute values differ from vendor to vendor. So it's very hard to know whether one vendor used an endocardial measurement and another vendor reports a mid-myocardial measurement. And if you just do a meta-analysis, as Dr. Gormley was pointing out, without knowing exactly what the metric was, we may end up with results which are not very accurate. So it's extremely important, and what we've done at our institution at least is for all the amyloidosis patients, we have one echo machine. So anybody with a amyloid diagnosis, they only get imaged on that machine, and there's only 2 sonographers who can scan them. This is very stringent, and I think it's very challenging to do. But for research, maybe that's what we need to do. We need to qualify certain machines at every institution. So you need to know exactly what machine it is. We need to qualify the sonographers, somehow improve the standards. Once we do all of that, I think we are in a much better shape.
And then my final comment before we just open it up, and I hope all of you will tee up your questions and go to the mic and just feel absolutely free to jump in here. This issue of variability and reproducibility. The things that I know that are important to multiple parts of the FDA when we start having these discussions are two questions. What is the within subject variability of your image in this case? And what ultimately, if you're trying to do a apprentice kind of surrogacy exercise, what's the minimally important difference of that change in the image that ultimately a patient cares about or can feel or detect? If your minimally important difference is smaller than your within subject variability of the method, you don't have a chance of separating out signal from noise in that situation most of the time. So Norman, please jump in here if you have other thoughts about this.
Yeah, I have. I'm really glad you brought up this issue because, in addition to the variability that's associated with the site or the machine or the sonographer or other factors, there will be for some kinds of outcomes at least. Significant true within subject variability and all of these things work against you as you are trying to build a case that my biomarker relates to something that you should care about. You can't do better than the noise that you have to deal with. That limits how much correlation you can get if your measurement isn't correlated to itself. It is worth every effort to do what you can to develop protocols for things so that you're not stuck with one sonographer and one machine that can do things. You've got to be able to, if you're going to collect substantial data, you are going to need to get a handle on all of these characteristics and limit them as much as you can. So I think that's a big deal.
Dr. Gormley, do you have any thoughts about that? And the reason, and what I'm thinking about here is I know that in your division, because of the nature of what you do, you probably get a lot of proposals for real-world evidence use. You can see in a process like this, rather than being drugs, it'd be like images. And these exercises that people go through in placebo borrowing from other places and that kind of thing, as far as how that those concerns of real-world evidence in drugs is relevant to this discussion, if you'd comment on that, that would be great.
Yeah, I think, I would echo what Norman mentioned earlier. And I think, just as well, some of the experiences we've had with real-world data, it's really sort of taking a first look at it and is it fit for purpose? Is it fit to address the questions that you're trying to answer? Oftentimes, the data source that is used can be really challenging to try to extrapolate the information that you're trying to answer. Whether that be claims data is really intended for insurance and things like that, and may not have the granularity of information that you need if you're trying to use it for other purposes. As well with EHR data, it also may not oftentimes have the granularity of data that you need, either even on the long-term, outcomes of interest. If you're trying to relate this to an EFS or progression-free survival or overall survival metric, not always having that granularity of data even there, let alone differences in imaging or other, biomarkers that may be captured differentially. And then also, real-world data has a lot of appeal because I think there's potential there in terms of, obtaining information in theory on larger, cohorts or cohorts that, may not necessarily be represented in clinical trials. And so there's a lot of strength and potential there. But the other issue that then I think really we struggle with is just, is it fit for purpose? And ultimately, sometimes when you start collating this data, there may be substantial amounts of missingness. And just because of either it wasn't intended to be captured or it just wasn't captured, systematically. And so sometimes once you start, looking at the data sources more closely and really assessing whether or not it's fit for purpose, you end up whittling down to relatively small numbers and unfortunately oftentimes substantial amounts of missingness. So it can be challenging to really use real-world data in some of the ways we'd really like to.
Right. And then I had one final comment before we'll just open it up to the floor in general, but one thing I wanted to be sure we got to Dr. Dorpala to comment on. Some AL patients, by the time they get seen, are really sick. And if you're going to try to do a trial on developing an image, if the risk of death is high, what are the problems with that? Because it seems like that they are the people who could most use a non-death outcome. And so does that change the approach of a development program for something like this?
Yeah, so Preston, you bring up a very important point. So in our longitudinal study, what you notice with these patients is the patients who die have died before they came back for their 6-month visit or for their 12-month visit. And the data that you have on change is on the survivors. These are the people who are alive at 6 months and 12 months, and of course improvement correlates with survival because you're seeing that. So the biggest challenge is how do we help these people who are dying early on whom we don't have information because they could not come back for the 6-month visit. So this is where I'm trying to put forth this proposal that we should be probably considering earlier time point imaging longitudinally to understand what happens. Like, we don't expect a change, right? We don't expect with plasma cell therapy, we don't expect a change within the first 6 months. But who knows? Maybe the persons who are dying, if you do an early scan, maybe we pick up something. And that's the most important cohort that needs to be helped. This is a cohort that we want to make sure. So at least for future trials, one proposal is to try to put forth early imaging markers, let's say baseline, and follow the oncology lymphoma model or something like that, and take a scan maybe 2 weeks or 4 weeks after initiation of therapy. And if there's absolutely no change or if there's rapid progression, you've caught that patient, at least for prognosis, if not anything else.
So, I was listening to the discussion around variability of imaging markers and thinking on the one hand, in this quest for the perfect imaging biomarker, are our criteria too stringent? Is there any example of an imaging biomarker in cardiology or other branches that really fits all this criteria? Everything has a certain inherent variability. On the other hand, there are examples where we have learned to work around the variability. For a good example is ejection fraction, right? It's one of the most, variable metrics, depending on which technique you use, which modality you use, and yet think about, every drug trial has used ejection fraction. A number of large device trials have used ejection fraction. I was thinking, for example, in the Heart Start study, the very definition of HFpEF is going to be based on ejection fraction. Yet it is one of the most variable metrics in imaging metrics in cardiology.
That phenomena comes up in a lot of things where you're developing imaging. I can tell you that there's a lot of very talented AI ML programmers looking at building histology platforms for computational reading of histopathology slides. And it's not common to get told as a sponsor that if on the trial that you're going to run, there's 5 people on the planet that can read these slides correctly. And so they all need to be on your event committee. Well, then you go and say, okay, well, these 5 people, they've been on other event committees. When you recycled cases back through the same histopathology and got the same, get a different reader to read it, say they got 3 of them. What is the inner observer variability between those 5 people that are the 5 people on the planet that can read these? This was a real conversation I might add. And the answer that I got was, it's high. And so I said, okay, well, then when you recycle the same slides back through and the same person reads them, what's the intra-observer variability? And the answer I got from that is even higher. So I said, so let me get this straight. These people are not agreeing with each other, and they're not agreeing with themselves, but this is the truth standard for training your algorithm. I struggle with that premise, but that's going, to your point, that's going on. And so what is the truth standard in these efforts to do these kinds of things? And I think that's a real struggle because we're assuming everything we have right now is perfection. Yeah.
Just a quick point. I think in everything we do there is variability. Probably death is the only thing that doesn't have variability in it. And the problem is that with imaging, it's just easier to show that. It's easier to show a modality, the variability within it, compared to 6-minute walk test, peak VO2, nobody wants to exercise people multiple times. So I think, everything has variability in it. I think in imaging, it's easier to show that variability because it's easier for us to actually repeat sometimes these tests and show that. But the same thing, we've done 6-minute walk tests in amyloid. The same thing essentially exists, that inherent variability. Heart failure admissions, we view them as hard endpoints. It depends on the geography and the region and the hospital practices, what defines the heart failure admission versus not.
And whether COVID is in the system or not, we saw major changes in heart failure hospitalizations and various trials as COVID came around.
Yeah, I see Dr. Dispenzieri has been waiting patiently. Angela.
Yeah, thank you so much. So a couple of points I just want to, so one point about variability and reproducibility, and yeah, it is difficult, but I want to remind us that, if it's for drug approval to get drugs to our patients, I mean, the peripheral nerve, the NIS score, Peter Dick went from country to country to teach each investigator how to do it. So, and the drug, patisiran and inotersen were approved for ATTR peripheral neuropathy. And so, again, there are limitations and there's variability, and sometimes you do have to, for the purposes of getting a drug approved, you may have to, do these outstanding things. That's point one. Point two is, in terms of the patients who are the sickest, the reality is you don't need an imaging endpoint in terms of drug approval because patients unfortunately are going to die early. So you have the gold standard, the sad gold standard, but you have that. So, as far as understanding the role of imaging and all the rest, it's of interest, but it's not keeping us from getting a drug approved. And the last point, and then I'll be quiet, is again, looking at real data and we talk about variability and perhaps slightly different platforms, this, that, and the other. Well, I would argue that in truth, if you have that inconsistency, in some ways it would be harder to prove a point because you have all that noise. You're not all pure and perfect. And so it feels like we're sort of, arguing that, well, it's too variable and that it's like, too restricted and whatever. So food for thought.
That's a very good point. And you're right. If you can show something through all that noise, it's probably even more believable. But I would imagine that developers who fund these programs look at that variability and question whether they're going to be successful or not, and it may impact decisions on whether things get funded or not, quite honestly.
I think it also depends on the question. If it's research, if it's for regulatory approval, or we're asking a mechanistic question, then obviously, you want, the key expert to train everybody in your imaging assessment or your symptom assessment. That way you get a highly valid result with as little variability as possible. But there's also this, counterbalance between scalability and then, something that is extremely precise, where we're trying to get a drug approved, for example, or learn about disease biology, or prognosis and natural history. And so it's just an interesting dichotomy here, because I think everything that we've discussed and everything we'll probably discuss over the next little while here in this discussion will be, kind of balancing those things. And I think it really gets back to what is the question we're trying to answer.
And just going to see if we've got online questions.
Oh, yeah, I've been kind of moderating the online questions.
Oh, okay.
Sorry to put in. There's been
you go ahead, because we'll get back to then the folks in the room. You go ahead and go.
Yeah, because there's been a lot of important and valuable comments and contributions in the online Q&A. A lot of these have been kind of touched on, a lot of comments from patients on how they were worked up and assessed. And I think a lot of the kind of emphasis here has been maybe on the clinical implementation of these technologies, which I suppose is a little bit beyond the kind of discussion of regulatory stuff that we're addressing today. But I think I'd like to comment on a lot of concerns that have been raised, that this is something all of us are working on, mainly with societies such as AHA, ACC, ASNC, ASE, all the imaging societies, in ensuring quality of implementation and accessing smaller centres. So that was just to kind of address a lot of the concerns that have come up online.
Anything on you? Okay, so I think we're good, and so we'll go ahead.
Yeah, thanks, Preston. This is Spencer. I think your point about drug developers being concerned about variability is, of course, a very good one.
Right.
And I wanted to, I was speaking to various people from industry here. There are a number of people that work in systemic amyloidosis who used to work in beta amyloid. So companies that are doing imaging, such as Dr. Stevens, or companies that are doing anti-A-beta drugs. And I think about 12 years ago, that group of people, including academicians, the regulators, sat in a room like this and had almost the exact same conversations. And what came out of it was something called ADNI, which was the Alzheimer's Disease Neuroimaging Initiative. And basically, every company who worked in the space provided funding into that, because they all knew that this is what was needed, because there were new PET imaging agents, there was new advanced MRI techniques, there were new things being done with lumbar punctures. Sort of the holy grail was to get to blood-based biomarkers. And now it's been going for 12 years, and it's really, certainly on the diagnostic side, really revolutionized diagnostics. And now we're seeing, as response to therapy, it's getting there, hopefully.
Yeah, we were talking about it earlier. I think it's 16 years, and they've just got refunded for their next 5-year grant, so it'll be 20 years that ADNI's been going on. And so it's been absolutely critical to the field of Alzheimer's drug development. Has really set the standard for the clinical instruments that are used, for the imaging protocols that are used, for the MRI protocols that are used. I think that probably the blood-based biomarkers couldn't be done without it. And it's taught us a huge amount about the natural history of the disease. You know, people can question, well, it's not a real-world population, but the amount of good stuff that's come out of it has been, I think, has been invaluable. And we were talking about it in the break, is that I really think that, and that's why I was happy to see the FNIH activity, something like that, is that we need to have big consortium trials where everybody gets together and agrees on the protocols and agrees on a process for an observational set of studies, so that we can define what the steps are and what we want to put in our trials, and then everybody agrees to it. And as was said earlier today, by Dr. Hoffling, is that there was a whole meeting yesterday about standardization of amyloid imaging for beta amyloid in the brain, talking about reproducibility, talking about metrics, talking about platforms, talking about software, talking about how to deconstruct where the noise was and where the important noise, and how you could improve the robustness of these trials so that you could really get quantitative data, which will open the door for therapy monitoring. So these things are going on in brain amyloid, okay? And we're probably 10 years ahead in brain amyloid compared to cardiac, but we should mirror that. We should look and take the lessons that are important and valuable from that space into this space.
Could I just ask one question follow on to that? So after 20 years of ADNI, what was the conclusion from yesterday's meeting about how close you are now to getting approval for any of those things?
It was a diagnostic meeting. It wasn't held by the therapeutic group. Okay. They explicitly didn't want to talk about therapy monitoring because it's obviously a sensitive issue within the FDA right now. But I think that it comes at an interesting time because we've just gotten, all of the three imaging agents have just gotten approval for quantitation as an adjunct to a visual read in Europe, and now the FDA is really looking about, does that make sense? And as we talked about earlier, as you go earlier and earlier disease, in the disease process, the accuracy of a visual read, which is how we're now approved, becomes less and less, and it really becomes critical that you have quantitation and you have quantitative metrics for the amyloid imaging. That, again, is exactly in parallel to what we're talking about today, is that anybody can read, an amyloid scan of the heart when you have somebody with highly symptomatic disease. But if you're going to go into secondary prevention, one, who are you going to screen, or who are you going to look at? And two, are you going to have the metrics to quantitate those that early disease?
Yeah, we're going to go to the microphone in just a second. Dr. Durala had a follow-up.
Just a quick question to comment on that. I think SNMMI is working with National Cancer Institute, and theranostics is rapidly growing. This is an area where I think we could collaborate with the imaging societies to get together people to work together and develop these. I think really great thoughts. ASNC could have an important role, ASC could have an important role, SCMR as well.
You've been waiting patiently. Please.
Yep, just had my afternoon cup of coffee, so I figured I'll try to bring some provocative questions to the group. My name is Michael Maitland. I've been working for Intellia Therapeutics for the last 2 years. But before that, I was sitting at tables like this as an academic investigator in industry consortia, trying to develop imaging biomarkers to improve clinical trials in solid tumor oncology. And I'm honored to be here. I thank ARC leadership for inviting us. And this is a phenomenal group. I've been reading your work for the past 2 years. You're really smart, high-quality work. And so you're bringing all that due skepticism to these, I think, kind of inside baseball issues about the imaging markers from an industry perspective. For the life of me, it looks like global longitudinal strain and ECV are far enough along that someone should be embarking on trying to qualify at least one of them as a surrogate biomarker for clinical trials. I think we'd love to have a surrogate marker for clinical trials. I think you would love one. I think patients would love one. So it begs the two questions. One is, what has kept folks, the really smart folks in this room, from embarking on that process so far. If resources were not an object, how long do you think it'll take putting all the efforts together in order to actually get to the point that perhaps one of those is a surrogate marker? And then just one quick comment is that shouldn't be the limitation, failure, because if you just start getting to work, you put together the infrastructure in order to try to get one of them qualified, even if that marker fails, there's an enormous amount of work that has been accomplished, infrastructure that's been put in place, and maybe one of the other markers that's following behind will be able to get qualified that much more quickly. So interested in one, how long do you think it's going to take, and two, why haven't we started yet?
I'm sure we'll have a lot of comments on this, but one thing that I'd like to throw out rhetorically to consider is if a trial like that fails, did it fail because the experimental image failed, or did it fail because the experimental drug failed? Okay.
Yeah, my comment's going to dovetail exactly with that. And I think we've, learned from, heart failure clinical trials about NT-proBNP and looking at the Prentice criterion as we think about that, right? There's no doubt it correlates with disease severity. It is incredibly prognostic. It's probably the sine qua non biomarker that at least establishes or perhaps gives somebody an idea of how they're doing and how they will do. But yet it has been elusive as a treatment target. So we know, if you look at a number of post hoc analyses from CVOTs, from heart failure therapeutics, that yes, you give somebody a beneficial treatment, their NT-proBNP or their BNP will lower over time. Now, what that doesn't mean, though, so that sounds good, and that's also very important patient-facing. You can show them their biomarker and say, look, great job. But one thing that has been established is if your NT-proBNP or BNP stays the same, that doesn't mean you didn't get benefit from the treatment. And so I think that dovetails with your comment, is it's very interesting, is that, if strain doesn't change, did you still do better? Anyway, so food for thought or building on what your comment.
Could I just address Michael's comment with some other provocative thoughts? I do think the time is right because the trials are getting bigger, patients are waiting longer to get drugs, and that's why we're all in this room, because we're all unsatisfied with where we think the field is headed. We have a lot of great compounds on the shelf, but could we get there sooner? To be honest, I think a lot of the impediment is not, two impediments I see. One is we're only going to be as good as our quote-unquote imaging phenotype as our clinical phenotype. So we all have to be willing to share clinical data as well. I don't think we're going to figure this out from just the picture. It's going to be in the context of everything else. And I take away that we need multimodal imaging. And I'm personally frustrated by how long it takes as an academic to get a data use agreement and share data with my colleagues. Columbia is not run by doctors anymore, it's run by lawyers, and it takes like 6 to 12 months to just share data that no one ever cared about before. But for some reason, I think someone thinks they're going to make a lot of money off of. Not that they are, but they think they are, and they don't want to share it. So that is a huge impediment to us being able to, I think, move the needle forward. And if anyone in the government can insist that, that data gets shared because that's concordant with what the patients want and what I think we all want, that would be a good thing.
Matt, one thing I'd like to understand is the major concern, the kind of that lurking, is it proprietary or not? Are we giving away the golden goose? Or is it the privacy concerns that say people at Columbia did not agree to let their stuff be mingled with Mayo and then transferred back and forth? Because what we're, in that guidance that came out from CDER, I think it was CDRH that put this out. When you look at these things like your iPhone, and it's collecting bioinformation from you.
Right.
And so there's Apple's privacy statement, but then it goes into a third-party software, and they've got a different privacy statement, and then it gets transferred over a network that has a different privacy law or standard. And so you end up in just the reality of doing this is you end up with like 5, 6, or 7 sets of privacy rules, and you then have to default to the most lax one see is it acceptable or not to your institution to protect the privacy of patients. And I'm having that issue with universities across borders. And so I'm wondering to what degree is that the driver versus just that proprietary concern for the value of the data?
I don't know. Someone else may know. My sense is they hide under the HIPAA, concern, but it's not really a big concern at the end of the day. The patients, I think, are willing to sign off and provide their data. So that's, I think, the biggest impediment that we have right now. It's not like this group hasn't, for decades, been collaborative and willing to put our data all in the same database, but it's becoming nearly impossible at some points in time. And I don't know why, but that's something I think has to get addressed.
And then final comment, would a federated approach solve that?
Maybe, where the data was still housed at the center and somehow, analyzed locally. But I don't know. Ashu, anything happening better on the other side of the pond?
No, I think we have the same issue. Sharing data is a huge challenge. I mean, we all have databases, and we've got quite a lot of data on these patients. I think it is absolutely logical that we share it and somehow come up to some conclusions about what it means. And having maybe some sort of resource to actually get through these regulatory barriers and allow us to analyze data would be very helpful. I think it may well be that it's hosted by one institution, and then that institution analyzes it and puts it out rather than hosted by a third party. But there has to be a solution to this.
Yeah.
I mean, if I can comment just quickly, me over here, is Andrew Stevens. It's again another sort of argument for something like an ADNI is that one of the real strengths of ADNI is that they build into the fabric of the contracts and into the fabric of the informed consent that this data will be publicly available, and then basically every scan, every piece of data is available on an independent server that people can download, and there've been I think over 14,000 papers now that have come out of the ADNI database, and so it solves that problem proactively. We have the same issue now. We're just finishing up a study called AMIPAD in Europe, where IMI, the European consortium, basically said you will make all of this data public if we fund you, and so it was built into the legal system right from the very beginning that's what had to happen, and I think it was hugely helpful.
Yeah, I think very similar to that. Like the heart share that we've just been shown today, like I've been trying to work with some of the third-party vendors that are working with NIH on echo analysis, and it's been over a year and we still don't have a contract signed based on IP and sharing our imaging. Again, masked and kind of HIPAA concerns, but really the concern is IP and licensing and stuff like that. So I think if there is a way for us to collaborate using a heart share kind of platform in that way, to use that to share our images in a more kind of accessible way, that you don't have to go through this quagmire of contracting.
I would agree. I was going to make the same points there. A heart share, I think, will in some ways solve a lot of the echo problems, because you could just reanalyze with AI and get your strain with one platform. I think with MR, it's more challenging because there are different ways to acquire the data. There are different sequences and different vendors. So we need another level of standardization before we can upload that information and have it reanalyzed. So I think that it's a two-step approach. You really need to have, at least on the MR side, I can say you definitely need to have some agreement about what technique, what system, what contrast agent, and then you'll be able to combine data.
So Dr. Michael Gerard-Sherrill may have some thoughts on that, because we were recently discussing about how to standardize between MR machines, and how do you standardize T1 mapping from one vendor to another.
It's still a challenge. We see it in some trial data we get. Even when you specify a protocol, some vendors may actually not give you the option to adopt the parameters which specify in the protocol. So there are, for example, vendors who just give you a menu of two choices, and there are other vendors where you can fine-tune things. So I think this will be overcome because there will be pressure on the MR vendors. Yes, it has helped actually ECV despite the optimism, has probably held it back a little bit because we're still dealing with these issues.
It's Meet Mitter from Mount Sinai, New York. It's actually picking back up all of this. With MRI, even our ECV calculations, and maybe others can comment here, is like we introduce error when we do those calculations. Whether it's a synthetic or an ECV based on hematocrit, if it's point of care, or a venous hematocrit, like what error do we introduce in the calculations? And if we're doing data sharing, different centers may approach it differently. I know one of my previous institutions was at, everyone got a venous hematocrit within 24 hours of their MRI. The institution I'm at now tries to use a point of care, and what does that do to our assessment? How does that inform response to therapy in a clinical trial, what we're assessing? And then down the road, when it's actually used as an endpoint for treatment in the real world, does that have implications? And do we then need a cloud server for the physician community outside of the clinical trial that's going to analyze those data for us?
I guess I can answer. Maybe Michael and I can answer the ECV question. It's a great point on hematocrit. We didn't even talk about that variable, but you need a hematocrit, preferably at the point of care at the time of your MRI scan, to calculate ECV, or you can do a synthetic calculation from the venous blood signal. But that's another important variable that we didn't talk about. And I also I'll ask actually, Michael, in the range of hematocrits available, it might be that when you have high or low hematocrit, you might see in, for example, AL amyloidosis with treatment. I don't know if that actually influences, and we don't have Mariana. I don't know if she's still on. Well, she's still on.
Mariana,
she can answer that question. Yeah. So I think it's an important variable.
And Mariana, I see you've had your hand up, please.
So our experience with the synthetic ECV is that it works well when you have the hematocrit within the range. So, for example, hematocrit within the range for clinical reasons, not for research, I would use synthetic, but I would never use synthetic ECV for tracking changes over time, for example, or for research purposes, tracking changes over time. And also, when we talk, and because CMR, of course, is much more recent, and T1 mapping is much more recent than ECHO, there is a huge amount of standardization, but even nomenclature and everything that needs to be done. For example, when we talk about ECV, we talk about a concept, but actually the calculation can be done in lots of many different ways. You can have ECV measurement by drawing lots of regional interest and actually averaging and everything, or you can have ECV mapping. You can have the co-registration done inline on the scanner with no human interaction. You can have a co-registration done with post-processing. So there is a lot to do in terms of standardization, definitely for CMR. But within clinical trials with a good core lab, we can control for lots of these aspects. And the progress we have made in the full automation of these processes, like for example, co-registration, motion correction of ECV mapping in the last 5 years has led to an incredible improvement in accuracy and precision, in my opinion.
Okay, well, we are almost exactly on time. We've got a few minutes remaining. Any other comments or things that somebody wants to ask about? Yes.
I'm just passing on an online comment that Cynthia Welsh made, that maybe the RDCA DAP may be able to help with the issues relating to data sharing. So I think this is probably a really useful resource for us to access, yeah.
For sure. Any others? Very good. So I'll end my session by turning back over to Isabelle. But just in doing so, I wanted to thank every single person who took the time to be here today, both in the room and virtually. If you look at the beginnings of our forum, they were humble, and a group of people who really just cared started working. And it has grown to what you see here. For all of you that have been here and supported this effort all the way through, thank you. And I foresee great things as this group goes forward.
Thank you. And from a patient perspective, when risk with these diseases is so high, we have to be mindful of the need to focus limited resources and collaborate. And I think today's meeting does just that and sets the stage for really promising further advances. And I would really like to thank everybody who's participated today. There's been a huge amount of work and thought and collaboration that has gone on behind the scenes to make today so successful. And in particular, Dr. Dorber for an extraordinary chairing of the meeting, and to Dr. Adigun, Preston, and of course to Kristen, who's really spent many months brilliantly refining the agenda and herding the headless chickens that we are sometimes to get us to produce our slides on time. And with that, I'd love to and with that, I'd love to hand over to Dr. Adigun for your final thoughts.
So thank you, Isabelle. I want to thank everyone, both in person and online, for being here today. I think we will all agree it's been a great discussion, great presentation. We started off the day highlighting limitations in animal models of amyloidosis. We've had a really broad overview of all the available imaging biomarkers. We've also discussed and identified areas that we can improve on by having need for purpose PROs specific for the area of amyloidosis. And we spent the majority of this afternoon talking about pathways moving forward. So I think a logical conclusion will be what are the next steps after we leave here. So I think we're going to take the totality of all the discussions we've had today back to the steering committee, review all the suggestions and identify all the things we need to work on. And from these discussions, we will identify work streams proceeding forward. But what I would like to encourage everyone here is the same enthusiasm that we've had today just to proceed forward, because we may be reaching out to a number of you to help us as we move forward to identify the best way to address these gaps and move forward with validating surrogates, improving what we currently have in helping our patients, and hopefully moving the bar forward so that when we come back, maybe in the next year or two, actually I think 6 months, because we'll be meeting again in June of 2023 with a forum focusing on TTR amyloidosis, that we can look back, assess how what we've done and where we're going, and identify what progress we've made and what else we need to focus on. But I want to thank everyone. It's been a great day. Thank you.